When you look at the structure of proteins, they have primary structure, secondary structure, tertiary structure, and quaternary structure. Depends on the folding of these proteins. And you want to star each of these and be able to explain them. Primary structure, that's the sequence of amino acids in the polypeptide chain. It's just a sequence of it. What amino acids are in what order. The secondary structure, this is where you have hydrogen bonds between amino acids within the same polypeptide chain, forms an alpha helix, and hydrogen bonds between adjacent polypeptide chains form what are called beta-pleated sheets. This is a beta-pleated sheet. You can see these hydrogen bonds between here. These amino acid chain can fold back on each other and form these hydrogen bonds connecting them, forming sheets. And then here is an alpha helix, where you see the hydrogen bonds in between the amino acids here of this helix. Tertiary structure, well, this is where that polypeptide chain bends and folds upon itself to produce a complex three-dimensional shape, such as this right here. It's folding back on itself. Well, if you take two or more of these different polypeptide chains and bond them together, like the tertiary structures, hook those together, then you get a quaternary structure. For example, hemoglobin consists of 4 tertiary structures bound together, and we call that a quaternary structure. Denaturation is a really important term. You want to make sure you star this, because denaturation of proteins involves the disruption and possible destruction of both the secondary and tertiary structure. Since denaturation reactions are not strong enough to break the peptide bonds, the primary structure, or the sequence of amino acid, remains intact. Denaturation disrupts the normal alpha helix, those beta-pleated sheets in a protein, and then coils it into a random shape. Where this becomes important is that when you look at enzymes and their active site, enzymes have an active site that catalyzes chemical reactions where substrates will come into that active site, and the enzyme will either hook these substrates together or can break them apart. If you denature enzymes, you're denaturing that active site, so it can no longer bind to its substrate. And it can greatly affect enzyme activity, permanently stopping its reaction or its function. Pause the video and answer the questions. Here are the answers. And that concludes this video, and let me know if you have any questions.